short video, we'll discuss parts of Chapter 4, Module 3, which covers brain plasticity. So the question is, what is brain plasticity? It's the ability of the brain to change. So our brain has this amazing ability to be plastic and to change in response to certain things. So when does this plasticity response occur? What did we talk about in the last chapter? Learning. So during learning, that response that we saw of the postsynaptic cell and the presynaptic cell, those changes that we saw in the dendrite and in the axon terminal, those were plastic changes. Those were actually changes that were occurring in the brain. We also see it occur in addiction. So there are changes. The drugs actually cause physical changes to the brain. What we're going to talk about in this video, the type of plasticity we're going to discuss, is in response to brain damage. Now, there are many potential types of brain damage. We could have tumors can cause damage to the brain. Various infections can cause damage to the brain. Exposure to toxic substances, whether it's something you ingest on your own, like a drug, or it's something that you're exposed to that you're unaware of, a noxious smell that you go by. Degenerative diseases like Alzheimer's, Parkinson's, Huntington's, all of those are damaging the brain. A couple that I want to talk about in a little bit more detail are closed head injuries. And so this is what it sounds like. This is where you have a brain injury, a sharp blow to the head, but it doesn't puncture the brain. So this is like you're in a car accident and you hit your head really hard. That would be a closed head injury. Or you're out there on the soccer field and you fall and you get a concussion. That would be a closed head injury. Same thing with football players. It's a really big thing right now with football players and this repeated head injuries that they come across, leading to something called chronic traumatic encephalopathy, or CTE. And this is where these individuals, after repeated head trauma, they suffer memory loss, mood issues, impulse control issues, and it often starts years later. And so some research I've looked at suggests that it's really not actually happening in the brain, but it's because, well, there's damage to the brain, but it's because that the cerebral spinal fluid, which takes away toxins out of the brain, can't clear that because there's actually compression on the spinal cord. It was an interesting study that I read. Another one that I want to bring your attention to is stroke. So stroke is basically a cerebrovascular accident. It's temporary loss of blood flow to the brain. So that's where it's blood flow being that cerebrovascular. And it's an accident because obviously we didn't intend for it to happen. And it's a very common cause of brain damage in the elderly. And it's important to point that out is that this plasticity response that we're talking about, it decreases as we get older. And so when you have this brain damage in the elderly, yes, they recover often, and that's the amazing plasticity response of the brain, but maybe they don't respond as easily as someone who is younger. And the older they get, the more difficult that recovery may be. So we're going to talk about different types of stroke. So if you look at this image, what does it look like here? This is a blood vessel. And in the blood vessel, you see an obstruction or a clot of some sort. So that's called an ischemic event. So where you have a blood clot or an obstruction of an artery, the neurons lose their oxygen and glucose supply. So if you have this obstruction here, the blood can't flow from one place to another. And so if the blood's flowing in this direction, anything that's beyond that clot isn't getting oxygen, isn't getting glucose, and if a cell's not getting that, they're not making energy, and thus they'll die. Ischemic events are the most common type of strokes, and they occur, they're about 87% of strokes. Now, what does this one look like? Here, it looks like you have blood going out into the actual tissue. So this would be considered a rupture or a hemorrhage. So you have a ruptured artery, so a blood vessel that's been ruptured, and the neurons that are flooded with these excess blood, calcium, oxygen, other chemicals, all sorts of things that normally that blood-brain barrier protects it from, or doesn't expose the brain to in such large amounts. So another word sometimes it's used for this is an aneurysm. So it's the ballooning. So an aneurysm is a ballooning of a weakened region of a blood vessel, and if left untreated, that aneurysm can rupture, leading to this hemorrhagic type of stroke. So following a brain damage, whether it's from stroke or something else, surviving brain areas will increase or reorganize their activity. Now, think about it. Maybe you've known somebody who has had a stroke, and if we think about the way the brain's organized, say it's their left motor cortex, and then that would mean they would lose the ability to move maybe their right arm. But over time, perhaps that ability recovers. And so that's plasticity. That's because the surviving brain areas are going to reorganize their activity to reinnervate those areas that were damaged. So some simple mechanisms that aid in this recovery after injury are the regrowth of axons and axon sprouting or collateral sprouting. So first we'll talk about the regrowth of axons. So when a cell body is destroyed because of a hemorrhagic event or because of a loss of nutrients like glucose and oxygen, they can't be replaced. Cell bodies, our brain doesn't multiply, their neurons don't multiply through mitosis. We have a very limited amount of stem cells and neurogenesis that occurs, but for the most part, cell bodies that die are not replaced. But under some circumstances, damaged axons can grow back. In particular, in the peripheral nervous system, a crushed axon will follow the myelin sheath back to the target. However, this is where it's kind of an interesting distinction, is cut axons may not grow back to the correct location. So crushed, meaning the remnants of the axon are all still there, they're all kind of intact, and they have the same pathway. But if you cut an axon, there's something different there, and it may not be able to grow back. And it grows in the periphery at about a rate of 1 millimeter per day, so pretty slow. So this diagram is just kind of trying to demonstrate that for you. So here, this is out in the periphery. So again, those are motor neuron cell bodies that are in the spinal cord that are going out to those muscle fibers. And here, we didn't talk about this. I told you it wasn't going to hold you responsible for knowing that the myelin in the periphery is made by Schwann cells, not oligodendrocytes. And so here you have this site of injury. Now, this area is going to degenerate because it's been damaged, and then microglia and other cells will do what's called phagocytosis and kind of gobble it up. And then it will regrow, following the path of degeneration. You have this growth, and ultimately you have the fiber connection that's reestablished. Now again, it's better if it's crushed rather than severed. So in the central nervous system, especially the mammalian central nervous system, axons do not regenerate. It just doesn't. There's lots of research trying to figure out why and how we can promote this growth, but it doesn't happen. So here's what we just saw in the peripheral nervous system. You have an axonal injury. The macrophages are going to phagocytose all that debris, and then there's this extending growth cone going back out to that muscle to reinnervate it. In the central nervous system, you have an axonal injury. There's no macrophages really to clear the injury. You have astrocytes that will come, and they create what's called a glial scar, really. And they promote the release inhibitory molecules like NOGO and various things. And it keeps this axon from being able to regrow. Now the question is, why do we have that? Why doesn't our central nervous system allow regrowth? And scientists work very hard to try to figure out that question and get around it. So why don't the mammalian central nervous system axons regenerate? As I mentioned, those astrocytes act as scar tissue, which is a barrier to that axon regrowing. Sometimes the neurons on either side will pull apart so that even if there was a potential of regrowth, they're not going to connect in the right way. And as you saw on the previous slide, those glial cells actually release chemicals that inhibit the growth. So again, that's what this is showing you here, is just right. And now I'm not going to hold you responsible for knowing those names. Just want you to be familiar with the idea that these astrocytes produce this glial scar that also releases chemicals that inhibits the growth. The other type of regrowth is axon sprouting, sometimes called collateral sprouts, which are new axonal branches that are formed by the non-damaged axons. And this is something that I studied a lot in graduate school, and unfortunately we don't have a lot of time to go into it. We call it reactive synaptogenesis. So here you have two cells that are innervating two adjacent areas. This axon is damaged, so it degenerates and goes away. And with axon sprouting, this surviving axon has axonal sprouts or collateral sprouts that then innervate the adjacent area. So you see here, it was just innervating this neuron. Now it's innervating both neurons. Here's another diagram from your textbook showing you here's that postsynaptic neuron, here's axon 2 and axon 1. They're both innervating here the same postsynaptic neuron. You get a loss of, there's an injury here and this degenerates. And so here you have these collateral sprouts to fill in and make up for that missing innervation. So you're going to also watch a video that talks about hemispherectomy. And so this is just kind of plasticity on steroids to me, it seems like. So it's removing half the brain, and it's a very radical surgery that's used to treat severe epilepsy. Now, it can only be done in young children. And that's because, again, as I said, the plasticity of the brain decreases as we get older. And so what you're going to see in this short video here is it kind of talks about the hemispherectomy, kind of shows what happens and shows how amazingly resilient this one little young girl was to having one of her entire hemispheres removed. And then if you want in the lecture notes, I'm not going to hold you responsible for it, but this video is Ben Carson is the one who brought this hemispherectomy surgery back in the 80s or 90s, I believe. And he was the first one to be able to make it successful. And so this is just a link to a documentary, if it's still active, that talks about his life and, where he came from and how he got to where he is.